Now we are going to study different experiments or the practical module of the microscopy. First of all, we will study in detail about what is the microscope and what are the different kinds of microscope which are available to us, and we can study the different microorganisms, different abilities of microorganisms, how to classify them, and how to identify the microorganisms by using the microscope, and what are the advantages of the microscope and what kind of the microscopes are available. So first of all, the microscope and microscopy. What is the microscopy? Microscope is an instrument used to see the objects that are too small to see them with the naked eye. Anything we use it which we cannot see with the naked eye, but we can use it through the microscope, and the microscope is used to see the any objects which cannot be seen with the naked eyes. It will be called microscope. So for the microscope, different little definitions are used for some kind of a microscope. What is the microscopy and what is the microscopic? So we will study them one by one. At what is the microscopy? The science of investigating small objects using such an instrument is called microscopy. The science of investigating small objects using an instrument, just we have studied that my instrument will be the microscope. It will be called as microscopy. And similarly, the microscopic. Any objects which is microscopic, which cannot be seen with the naked eye, and we have to use the microscope to see any objects, will be called microscopic. Microscopic means an invisible objects to the naked eye unless aided by the microscope, unless we have to take the help of the microscope to see that specific objects, specific microorganisms will be called as microscopic. So we need to see what is a microscope. We have seen we have taken the picture of the simple microscope, and we can see the different objectives which are used for the simple microscope. And where is so you can see from here, it is the microscope. This will be the ocular lens of the microscope. From here, it is also called as eyepiece. You have to see with your eyes, and you have to put your eyes there with the eyepiece and ocular lens. It will show you the microorganism or with the object you are going to see. This is the ocular lens, and the line of the vision is there, whether it is magnification, what kind of magnification or resolution you want to see it. We will see it there. Then the body tube. This has the body tube of the microscope. Similarly here will be the body tube or path of the light. The body tube will goes out through the path of the light in that is reason, and then there is the prism which will reflect it, that the light towards the object where we want to see it. Here it is the arm of the microscope, and then the objective lenses. Here are the objective lenses which we can put it at to 10x or 20x or 30x or 100x's. And then the stage, it is the stage where you will put your object there to see it. And then the condenser will focus the objects which are different lights are giving, and the prism is just giving the lights to different directions. So condenser will be focused. Condenser is going to focus it. Similarly here, the body tube and then the condenser lenses are here. And then the diaphragm controls the amount of light entering into the condenser. Then an illuminator, the light source. You can increase the light source and you can decrease the light source according to the illuminator and the light source, and the base with the source, and the coarse focusing the knob, base, fine focusing knob. There is a difference between the fine focusing knob and the focusing knob. It will be more clear and more better if you are going to. It depends on the eyes of the. It varies from the person to person, and you can also use the fine focusing knob to see the objects clearly. So these are the different parts of the microscope, which is just a simple microscope. Next, we will also study the different other kinds of microscope, and which are the different materials which are considered inside it or not. But this is just for the understanding the simple microscope studies.